Hello everyone, my name is Amanda Tolk from Xstrahl. Welcome to our clinical education webcast. I'm pleased today to welcome Kurt Byrnes, a radiation oncology medical physicist from Just Skin on the Sunshine Coast of Australia. Kurt will speak today about Grenz radiation therapy commissioning for medical physicists. Grenz rays are defined as up to 20 kV. Due to the ultra-low spectrum of the X-ray beam, they have historically been used for the treatment of benign dermatological conditions when other therapies have failed. In recent years, there's been a resurgence of interest in Grenz rays and their clinical applications. Today, Kurt will provide an overview of his experience of commissioning Grenz rays on the Xstrahl X100 system following the AAPM TG61 protocol. He will cover protocol comparison, half-volume layer measurements, percentage depth doses, and measurement setup. And then we'll conclude with lessons learned during the commissioning process. There will be a live Q&A immediately following the presentation. Thanks again for your attention. And with that, I welcome Kurt Byrnes to today's discussion.
Thanks, Amanda. A few years ago, I was working at a large hospital and I met a dermatologist who asked me about Grenz ray therapy. I couldn't answer the question. Even when I looked online and in my physics textbooks, I found very little. Here on this slide is the entire entry on Grenz ray therapy that I could find in my textbooks. Now, why am I telling you this? Because fast forward a couple of years and I'm now working at Just Skin doing superficial therapy between 50 and 100 kV. I began working directly with the same dermatologist, and when we looked seriously at delivering Grenz radiation, the lack of information made this seem daunting and overwhelming. My goal in this presentation is to show you that commissioning a Grenz therapy service isn't more difficult than commissioning any other energy. So what is Just Skin and who am I? My name's Kurt Byrnes and I'm an Australian medical physicist who works in radiation oncology. I've been specialising in superficial photon therapy at Just Skin since 2021. Just Skin is a group of small clinics that treat non-melanoma skin cancer in Queensland, Australia. Following the first site opening in Maroochydore in 2016, clinics in Gympie, Noosa and Toowoomba have opened and are treating patients. All of these sites use the Xstrahl 100 machine with field sizes from 1.5 centimetres to 18 centimetres. Until recently, each machine treated using only 50, 80 and 100 kV. We've been treating Grenz patients at our Noosa site for over a year now. The two main diseases we treat are field change with actinic keratosis and disseminated superficial actinic porokeratosis, more commonly known as DSAP. Before I get stuck into the technical details, I want to highlight the key messages that I want to deliver. Firstly, and most importantly, working with any uncommon treatment type like Grenz radiation, reach beyond your usual network of physicists. Xstrahl can put you in touch with other physicists globally who can help. Even though there was very little published information, there are other physicists in the world who know what they're doing. I have found complete strangers to be very generous with their time when helping me set up Grenz radiation. The item that produced the biggest delay for us was finding an external physicist to do a dosimetry audit. This is a legislative requirement here, and I would highly recommend finding someone independent to redo the absorbed dose measurements, even if it is not legally required where you live. This can be very difficult if you're the first in your region to set up Grenz radiation, and I don't have a good solution for you, but this is something to start considering very early on in the process. One of the biggest consumers of my time in this project was testing different materials to use as shielding and cutouts. This became such a large and ongoing project that I won't be going into specifics in this presentation, but the take-home message is that the atomic number of the shield has a big effect on the attenuation. We all know this when we talk about lead and tungsten with low energy photons. But with Grenz, I've seen some huge differences between carbon-based bolus that's used for MEV electrons and oxygen-based water. Whether using measured values or calculated values, the size of the cutout makes a negligible difference, so only an inverse square law correction is needed. This may not be the case for very small fields less than 4 centimetres. As you'll see in later slides, I've taken HVL measurements at two different source to chamber distances, which yes, I did get quite different results, and no, it does not make a huge difference to the final dose measurements. For a physicist looking into any unusual treatment type, the first thing to get right is the dosimetry. Unfortunately, Grenz radiation is not in wide use in Australia or globally, so little information existed in the literature or within the community of physicists that I usually turn to. The Australian College of Physical Scientists and Engineers in Medicine has published recommendations for superficial therapy called the Australian Recommendations for Quality Assurance in Kilovoltage Radiation Therapy. My work was based on this document, which I highly recommend for physicists, even those based outside of Australia. As per these recommendations, I use the AAPM Task Group 61 protocol. I predominantly used a PTW 23342 thin window ionisation chamber, which was calibrated in Melbourne, Australia by the Australian Radiation Protection and Nuclear Safety Agency. They were able to calibrate my chamber down to 10 kilovolts. I repeated the absolute dosimetry using the AAPM and TRS-398 protocols. As you can see, I got very similar results using each protocol. This was a great sanity check, and getting my head around the other protocols was a great learning experience. There is a recommendation document by the Swiss Society of Radiobiology and Medical Physics, which I highly recommend for anyone wishing to use the TRS-398 or German DIN protocols. Here are some photos of my measurement setup, both in-air and in-fender measurements. The equipment shown in these photos is the same as used for other energies. The in-air method has the advantage that the exact same calculation as the higher energies can be used. However, alignment of a thin window chamber in air is much more difficult than a cylindrical chamber, as a small rotation in the chamber results in a distance offset. Meanwhile, the in-fender method has the advantage of being a much easier and reproducible setup. Australians and Americans will be familiar with this formula from TG61 to calculate the dose to water at the surface of a phantom. This can seem like the scariest part, but I hope you'll believe me when I say that it's very simple. M is the electrometer reading corrected for temperature and pressure, as with most radiotherapy measurements. NK is the calibration factor taken from the ion chamber's calibration certificate. This should be corrected if the measurement distance differs from the calibration distance, which will be explained in the next slide. When interpolating between NK values on the calibration certificate, be sure to use the HVL obtained using the correct SSD. In my case, this was 30 centimeters. BW is the backscatter factor taken from table 5 in TG61 from the IAEA website. Be sure to use the HVL obtained using 100 centimeters SCD or the appropriate distance. The stem can be assumed to be 1, which makes life easy. The new TRS-398 has tables of geometry factors, and if you look at the 10 and 20 kV energies for this ion chamber, you'll see the same result. The mass energy absorption coefficient ratio can be taken from table 4 in TG61 or from the IAEA website. In my case, I use the HVL values from 100 centimeters SCD. A mistake I initially made was not correcting NK for a difference between my calibration SCD and my measurement SCD. When there is a difference between the reference point and the effective point of measurement, there is a dosimetry error when the chamber is used at source detected distances other than the calibration distance. Because my calibration distance was 30 centimeters and my measurement distance was 15 centimeters, this was a 0.7% error. This is easy to miss as most physicists are not used to doing reference dosimetry with a thin window chamber. In my case, this was picked up by my good friends at the Australian Clinical Dosimetry Service, who performed a secondary audit of my measurements. The Australian recommendations mentioned earlier give a great description of this correction if you need more information. There are several lessons I learned through this process, and hopefully I can save you some time and hassle. Firstly, don't bother using a pharma chamber. I had mine calibrated down to 10 kV, and the calibrations lab told me not to use it because it would be inaccurate, which I have experimentally validated by getting unusable results. If you're unsure about your measurements, start with the largest field size. This will be more forgiving to setup and calculation errors. It will also be the main field size you use when treating patients. You can see that each dosimetry protocol delivers similar results, so use whichever one you're comfortable with or have the equipment for. I performed half-value layer measurements in the usual way. The only thing I did differently was that I performed the measurements at 2 different source to chamber distances. For higher energies, like 100 kV, each distance yields the same results. However, at these low energies, there is attenuation by air, so the distance matters. Because my ion chamber was calibrated at 30 centimeters, I used the 30 centimeter results to determine NK, and the 100 centimeter results for everything else. The main challenge when performing these HVL measurements is that, like me, you probably don't have aluminium sheets thin enough. I bought a set of sheets with thicknesses from 1 millimeter down to 0.005 millimeters off AliExpress, which I measured the thickness of using a micrometer. My final results used a 0.05 millimeter sheet as the thinnest sheet and corrected the result using equation 5 in the Swiss recommendations. These are the HVL values from the latest machine I've commissioned for Grenz. You can see a pretty significant variation between the 30 centimeter and 100 centimeter measurement. If you decide to only use the 100 centimeter HVL, the effect on the final dosimetry in my case would have only been 0.2% for the 10 kV beam, so it's not particularly significant. I'm going to continue doing the HVL measurements at both distances for energies 50 kV and lower. However, even a 10 or 20% difference in HVL makes very little difference to the final dose. Because I don't have the equipment to accurately measure the thickness of a 0.01 millimeter filter, differences are arguably within the uncertainty of my filter thickness. Unless you have high quality measurement equipment, I don't recommend using sheets thinner than 0.05 millimeters. However, I did find that my cheap sheets from AliExpress gave consistent results to my expensive sheets when I used my own measured thicknesses. Here is a plot of the 20 kV percentage depth dose. As you can see, the dose drops to 50% at 1 millimeter. This presents quite a lot of challenges when my thinnest piece of solid water is only 1 millimeter thick. What I really needed was a thinner material that is water equivalent. After trying out a few materials, I found that 0.1 millimeter thick PVC sheets gave a curve within 2% of my standard imaging solid water. These also match very closely to the BJR supplement 25 data, which is also plotted in this graph. It's worth remembering that these PDDs are mainly used by radiation oncologists to help with energy selection. Like me, most physicists were trained on Linux, where the accuracy of the PDD is critical. This is just not the case for such low energies when prescribing to the surface. So don't lose sleep over this, and don't waste your life measuring PDDs. These measurements were the most time-consuming, however, spending so much time added little value. The BJR data was quite accurate for 20 kV and a great starting point and reference for me. I tried making a custom water tank and using an agent that destabilized the meniscus in order to reduce the minimum depth. I managed to get the depth from 3 millimeters down to 0.5 millimeters, but the depth was not consistent and not thin enough to be worthwhile. If anyone has a method to use water, I'd love to hear it. The profiles for the largest field sizes were the most heavily scrutinized. The heel effect was not apparent on field sizes 10 centimeters and below. The results in the next slide were taken using a piece of EBT3 film with a 3-minute beam delivery. The only practical change this made was for the therapists to direct the anode end of the beam to overshoot the patient where possible, which is good practice for all energies when using large fields. This table gives the size of the field above the certain percentage of the central dose. When looking at the size of the field above 80% of the central dose, you can see the size of the field is within 3 millimeters of the desired 150 millimeters. However, when looking at the 90% values, you can see that some of the field sizes are below 140 millimeters. This means there is approximately 1 centimeter wide area where the heel effect has reduced the dose to 90% in some very specific parts of the field. When using smaller fields, like we have typically used to just skin, the heel effect is difficult to find. However, when using fields 15 centimeters and greater, it became evident. I should also add this data is only from one site, so it cannot be assumed to be present in all Grenz sites. Most of the practical issues within the department surrounding the use of Grenz have actually been issues with using the larger field sizes, as we have rarely used the 15 centimeter cones in the past. This whole process took about a year for the first site, but as you can imagine, the second machine was done in a similar amount of time to any other energy. One of my main long-term goals for all energies, especially Grenz, is to determine full uncertainty budgets. This will guide the allocation of my time and resources for future QA and commissioning work. As highlighted, measuring these PDDs presented many challenges. I'm hoping to implement the use of gap chromic film in order to measure PDDs and small cutout factors for Grenz radiation. Although there was a measurable timer end effect for Grenz, I would like to explore the different methods of calculating timer end effect before finalizing my values. Thank you for listening, and thank you Xstrahl for giving me the opportunity to give this presentation. If you have any further questions or would like any of my measurements, please reach out.
Thank you, Kurt, for your presentation. If anybody has any questions, please do post them in the chat. We do have some already, so Kurt, if you don't mind, I'll go through those. Why did you choose to start treating with 20 kV as opposed to 30 or 10, or any other Grenz rate energy?
So, well, with 30 kV, we decided that anything we could treat with 30 kV, we could probably treat with 50. And we did look, and so our machines are set up for 10 kV, but we just decided to start with one energy to begin with, and definitely considered 15 kV as an option, but because my calibration certificates included 10 and 20 kV, we decided to use them.
Yeah, that makes sense, especially if you've got the certification for it. Why do you not recommend the pharma chamber? Just that's the question that's come in.
Yeah, well, there's a few reasons. A lot of it comes down to the wall thickness of the pharma chamber. You know, if you could accurately calculate all the perturbation factors, you might be able to use it. But because of the low penetrability of these energies, the wall thickness has an effect. The fact that it's curved means the perturbation factors are hard to calculate and have high uncertainties. Also, because you've got such a large volume in the beam direction, you get a lot of dose volume averaging. So potentially your effective point of measurement is not at the center. So you end up with a result that you can't trust. Whereas the good thing about pharma chambers is they're very consistent across the whole spectrum of energies. But I found not to be the case, and that's what I was told would be the case.
Okay, so secondary to that, which chamber would you recommend for somebody who's about to start commissioning Grenz rays on one of their devices?
Yep, well, the PTW 23342 and the 23344 are very good. And I was lucky enough to have one of those because a lot of the literature out there uses both of those. So the AAPM documents, the new TRS 398 has those geometry factors. But apart from that, I would really like to try the Xstrahl X100. That's meant to be used for the 50 kV intraoperative breast treatments. It's got a large volume and a thin window with no stem effect. I think that one would work really well in air. And I know some of the other, Xstrahl has some other thin window chambers that not many people in Australia use, but I think they would probably work well.
Okay, so some future work if you're going to commission extra sites then.
I guess so. We're up to 2 sites now. We've just started treating Grenz at our second site, and if we were to keep doing sites, I guess there's all the usual extra things. Once you've commissioned the machine, getting someone else in to audit your measurements is always good practice, and that can be difficult. Setting up your patient-specific QA. So in Australia, we always do a secondary calculation, which is just a correction-based calculation that we use, and all the documentation that you would normally do for any energy. I know for us here, we didn't usually use the larger cones, so the 15 centimeter and the 18 centimeter cones. So that was, I would say, the biggest thing for us. For everything else, practically there's not much difference between Grenz energies and these higher energies, but the fact that we moved to using bigger cones, that had, you know, a lot of something we just weren't used to. And I guess I'm not sure how it works in all the other countries, but definitely some billing issues, like certain you know, like Medicare type billing or health insurance billing may not cover, you know, everything for Grenz. But
I think it's a resurgence of interest in Grenz rays, and how it's billed against how it was used historically is going to be very different. So definitely, from my opinion, it's great that you're commissioning these energies again using current standards and current dosimeters that are available, because everything else is very historic. Let's have a look at what other questions have come in. So you've mentioned you're going clinical with Grenz rays, but what were the steps to going clinical? I mean, how did you instigate the QA or the clinical service?
Well, I guess initially, you know, a lot of literature review. This was something that, you know, was bubbling away in the background at work for a long time. You know, you have to, I guess, cross all your T's and dot your I's when doing something like this, just because hardly anyone else is doing it, you know, and shooting off a lot of emails to people I know saying, you know, do you know people who've done this? Luckily, all of my, say, routine QA is, if you've got a thin window chamber, the routine QA is exactly the same. So, you know, you just do your output factor measurements and half value layer measurements. As I said in the presentation, PDDs is a bit of a drama sometimes. But I would say you don't need to overthink the dosimetry. Just use your protocol and have someone double check your measurements, because I found them all to be pretty user friendly, mostly because say your P stem and your polarity, your recombination, all these factors are just one. So that's really what took me a long time to set a lot of this up. I wanted to make sure that, you know, you look into where am I going to find all these factors? Am I going to have to simulate these in Monte Carlo? But the reality is, with these really low energies, it's actually really easy, because a lot of these factors are just one, and, you know, your cutout factors, they're basically one as well. You just have to correct for inverse square law.
So you mentioned there aren't many other centres doing Grenz rays, but have you found any allies in commissioning at all? And I think for this audience specifically, it'd be great to know if you could be like a contact point or a reference point for somebody who might be considering doing Grenz rays on their machine with Xstrahl 2.
Yeah, for sure. Yeah, just get in touch with Xstrahl. They'll give you my email, or send me a LinkedIn message or something. Definitely happy to help. Yeah, so who else does it? Yeah, so I had a bit of help from, I don't know the hospital's name, but one in Switzerland, Jerome, helped me out a lot.
Zurich,
and,
yeah.
I don't, there's another site that does it in Sweden somewhere. Is it in Malmo maybe? Somewhere in Sweden does Grenz?
Yeah, Malmo does.
But I don't know where else does it. I don't think anywhere else in Australia does it.
Well, hopefully after today's presentation, we might collate who else might be considering doing Grenz rays, Kurt, because I think it's a really good resurgence of interest in benign dermatology treatments. And just to just cover off, what clinical conditions are you doing? I know you had it in the slide. I think that's what you're doing currently. Where are you hoping to go with this? I know you're the medical physicist, but you might have insight.
Yeah, I mean, I think we are sticking mostly to those two conditions. I think our next steps that are really to treat larger areas, you know, people have whole legs covered in this, you know, both arms, you know, and we're really limited to our 18 centimeter cone or 15 centimeter cone. So I guess our next step is not so much, you know, treating every disease under the sun. It's more like, can we treat, you know, different sites on the body, and a lot of it is, you know, the staff, the doctors, me, the therapists, we're not used to doing things like this, you know, doing these large areas. So yeah, a lot of it is it is very, you know, it's very safe, it's very low penetrability, but we do still have to be very careful.
Of course. One other question that's come in, which is very physics orientated. Why do you think the heel effect is not evenly distributed, has come in? And of course, that would be important if you were going to match those large fields together, given you've got long limb extremity that you need to cover.
Yeah, that's a good question. I think perhaps it could be a few things. Like and to be honest, the heel effect was a lot smaller than I thought it was. I think potentially if you've got, you know, depending on your setup angle, you've got a tiny bit of slack in your applicator. That normally doesn't matter, but I think maybe for these large field sizes with this small, low energy, you know, that sub-millimeter bit of slack might make a difference. But with my other two sites, so I've done this with Grenz at Toowoomba and another site, I didn't see this heel effect almost at all. So it is something I want to keep doing. I've only done it, say, once or twice during the commissioning, but something that I would like to repeat once a year or more often, and hopefully it's consistent. But it was unusual. As you could see in that drawing that I put up, it did go to the side more than I was expecting, and only to one side. So it could be my setup error as well. So I'll try it again later this year, and I'll let you know if I get the same thing. Okay,
perfect. So Kurt, thank you very much. Great to discuss your presentation, and thank you for sharing your presentation today. We're now coming up on our time, and I want to thank everyone for your attention today who attended, and please do feel free to share or reach out to Kurt or I if you've got any further questions. And with that, I will close the webinar for today. Thank you everyone for attending.
Thanks everyone.
Thank you.